Okay, I think we're getting that started. So thank you all for coming today to this seminar for the Norwich Institute of Healthy Aging. So you might have noticed that Anne-Marie is not here. Anne-Marie is away. She's at the Hay Festival. So I wish I was there, but there we go. So you have the pleasure of myself chairing today. If you don't know me, I'm Michael Homburger, Professor of Applied Dementia Research at the Norwich Medical School. But it's my great pleasure to introduce Professor Rik Henson, who is Professor of Cognitive Neuroscience at the University of Cambridge, and also a co-lead of a study called the CamCAN study, which he talks today about, which is really interesting, looking at longitudinal changes for lots of different variables, but he will talk mostly about cardiovascular disease today. So without further ado, actually just etiquette, so please keep your video and microphones off during the talk. You can type questions in the chat box, which we address then later, or you can come on video after the talk to ask your question in person. But without further ado, I just hand over to Rik. So welcome, Rik, and the floor is yours.
Thank you, Michael. Let me just share my screen. Can you see that? Great. Yeah, so I'm going to talk about late life cognition and how that's affected by midlife activities performed 2 or 3 decades earlier and current cardiovascular health, using some data from the CamCAN study, which I'll say more about in a moment. To start with, this is a horizontal section through a T1 weighted MRI of a 19-year-old individual. And I'm going to show you the brains of different people of ages 19 to 88. And as you can see, as the age goes down the left, as you get older, the grey matter with the ribbon of slightly grey tissue around the edge of the brain tends to get smaller. The white matter that connects different parts of the brain also shrinks. And as a consequence, the ventricles, these areas of dark tissue, expand. So if I now show you the 19-year-old on the left and an 86-year-old on the right, you can see the dramatic effects of age on brain structure. Obviously, MRI hasn't been around long enough to track the same individual over the whole adult lifespan, but these have been roughly matched in shape. And this is a very typical profile of healthy aging. We also know that cognition changes as you get older, or at least some types of cognition do. Fluid intelligence and memory decline quite rapidly, language and semantic memory less so. What's interesting there is you look at longitudinal data. So this is from a large number of European cohorts, and each participant has 2 data points joined by a line. And this is episodic memory Z-scored on the y-axis against age. And you can see there's a general decline, but there's a lot of difference between individuals here. So some individuals show quite a precipitous decline in late life, whereas others do not. And you can contrast this with the volume of the hippocampus. The hippocampus is a brain region that we know is important for episodic memory. And that shows a fairly universal decline in all people. Now, obviously, some of the variation in the episodic memory on the left could be measurement noise or be caused by other variables. But the general pattern that's been observed quite a lot is that some people, like this person here, can maintain their cognitive ability despite brain structural change, whereas others do not. And that puzzle is called cognitive reserve. So this is a term originally invented by neurologists who look at a brain scan of 2 individuals, both of whom might show evidence of aging or neurodegenerative disorder, and yet one of them was functioning normally in everyday life and the other was not. And so the former would be attributed this concept of cognitive reserve, which is really a ragbag term for something we don't really understand about their brains. Perhaps it's their brain function that's maintained their cognition despite similar structural change. And so I'll be talking about cognitive reserve later. What's important is to know what factors contribute to high cognitive reserve, and one of those we've known for a long time is education during youth. But this talk will focus on more modifiable factors such as midlife activities or cardiovascular health, which is easier for a government to nudge people on than, for example, changing education. And it's important to maintain cognition in late life because obviously the world's a complex place, particularly ability, for example, to navigate the internet and complete tax returns online or manage a bank account all requires good cognition in old age. So if we can hopefully maintain that for longer, we can be more self-sufficient for longer, and hence extend the useful periods of independent living. So today I'll start by just very briefly introducing the CamCAN cohort, because all the studies I describe, bar one, use data from this cohort. And then there's basically two parts to talk. I'll talk about midlife activities using a retrospective questionnaire and how they seem to relate to late life cognition. And then talk about cardiovascular health and its relationship between brain and cognition in late life. So the CamCAN cohort, the background of this is that there are many studies of aging using brain imaging and cognitive measures, but they tend to have small, unrepresentative and skewed samples. So for example, people who respond to adverts who tend to be the super healthy aging, for example. So we wanted to recruit a more population representative sample of adults from 18 to 88 and acquire cognitive brain data, that's MRI and MEG, genetic and lifestyle data on this sample. And hence CamCAN was born in 2010. This is our main website if you want to read more, and we were funded by the BBSRC initially and by European ERC money subsequently, as well as MRC money at the CVU in Cambridge. So you can read more about the protocol in this paper by Shafter et al. On the bottom right of the screen. But briefly, we actually recruited people from GP surgeries using an opt-out procedure. So we actually knocked on their doors and they had to say no if they didn't want to take part. So we've got about 35% compliance. So I wouldn't claim it's perfectly population representative, but it's much more representative than many other cognitive neuroscience studies. If they agreed, they did a 2-hour home interview where we asked questions about their health and their lifestyle, etc. And then we selected a subset of 100 per decade from 18 to 88 years of age, so about 700 people in total, who came into the lab on 3 visits to do, over those 3 visits, in total about 7 hours of cognitive phenotyping, an hour of MRI, and half an hour of MEG. We reassessed their cognitive abilities online much later, and we're currently rescanning them. So we're acquiring MRI and MEG data nearly, well, over 10 years later. So this will be a very valuable longitudinal brain data. We've also made the data available to researchers on request, subject to basic agreement to maintain confidentiality. If you want to use this data, please do apply. It's a very easy web process on this website here, and we've probably had over nearly 2,000 people researchers around the world who've actually used the data for research. I know there's a special interest in Norwich on diet. There is some diet information in the CamCAN data set. It's probably not sufficient for fully qualifying for Mediterranean diet information or whatever, but if you are interested in diet information, we do have that, and no one to my knowledge has really used that in the CamCAN sample to relate, for example, to brain health. We've probably published, well, there's probably over 100 publications now using the CamCAN data, and you can look at this link at the bottom if you want to look at any of the publications that I'll talk about today or any other publications. So I'll be just talking about a fraction of studies that we've done with CamCAN. I wanted to focus on lifestyle and cognitive health because Michael said that would be interest to you guys. And all the data I'll talk about come from this 700 people with tested in 2011. So the first part is midlife activities. So ideally, of course, we would measure lifestyle and cognition in midlife and then see how they predict cognitive abilities or health 20 or 30 years later in late life. So we do prospective studies. But of course, those studies are very hard to do, to get funding for and to run, particularly with MRI. So what I'm going to talk about is retrospective studies. So we scan people in their old age and we ask them to describe their lifestyles early in their life, in mid-age and in youth. Of course, there are caveats to this, which I'll come to later. And in fact, I'll talk about 3 studies, the first of which is from another cohort, the Lothian birth cohort by Gao et al., and then 2 from CamCAN. The first of these by Gao et al. Just was the first, I think, to find that midlife activities might be particularly important for old age cognition. And so it's easy to find correlations between lifestyle and cognitive ability, but the direction of causation is always unclear. So does lifestyle affect cognition? So people who have healthy lifestyles might have better, healthier brains and better cognitive abilities, or do cognitive abilities lead to more healthier lifestyles? It's hard to get around that, but to solve that problem of causality, but one attempt was made in the Lothian birth cohort, because these are people tested in their 80s, but they also had cognitive data from childhood. When they're 11 years old, there was a national school test done, and they found the data for that. And so these people we can control for their childhood cognitive ability. Anyway, they were asked to retrospectively judge their lifestyle according to 3 life periods: 20 to 30, 40 to 55, and 60 to 75. And therefore, you can ask whether midlife lifestyle affects current late life cognition, even after adjusting for early life cognition. And so this is more about the Lothian birth cohort if you want to read. But in this study by Gao et al., there were about 384 people aged 83. And their lifestyle activity included physical activity and social intellectual activities. Their measures of cognition were verbal fluency, story recall, etc. So measures of including fluid intelligence. And you can take a first principal component of these if you want, and you'll find they load on Spearman's G. And of course, they also adjusted for biological sex, number of years education, social class, smoking and alcohol, etc. But the main finding was that midlife social intellect activities were significantly related to current cognitive abilities, even after adjusting for all these covariates and their early life cognitive abilities. They also found that late life physical activities were associated with current cognition. So these 2 findings are the basis of this talk. So the midlife activities and the late life physical activities are something that we also followed up in CamCAN. But this, to summarize this Gao et al. Study from the Lothian birth cohort, midlife social interactivity appears to be important for late life cognition, but this is only behavioral data. They didn't have any brain data. And to really find evidence for this concept of cognitive reserve that I introduced earlier, you need to show that these lifestyle variables moderate the relationship between cognition and brain. So cognitive reserve refers to the fact that your cognitive abilities are better than be expected from your brain. So to test that, you need to have brain data. And that's what we did in this study by Chan et al. From the CamCAN cohort, led by Dennis Chan. Again, the PDFs for all these papers are on the CamCAN website if you want to read more. But we took 205 people in the CamCAN cohort who were retired, over 65, and we used a standardized questionnaire called the Lifetime Experience Questionnaire, or LEQ, which measures a number of things, which I'll explain in a moment. And we assess cognition by the Cattell test of fluid intelligence, but we get the same results whether we take the first principal component of a large number of cognitive tests. And for brain structural health, we use total grey matter from a structural MRI. This is what the neurologists would use in a clinic or some eyeball estimate of this when they come up with this term of cognitive reserve, the fact that some people can maintain cognition despite structural brain damage, and they usually look at grey matter. So total grey matter adjusted for head size is our measure of structural health. So this lifetime experience questionnaire, the LEQ, involves a large number of questions which can be divided into 3 life periods: young, adult, midlife, and late life. And within each of these 3 time periods, there are 2 types of questions. Questions that are specific to that lifetime period. So for youth, that's largely education. For midlife, that's largely occupation. And there's another set of questions, the non-specific activities, which are the same across all 3 life periods. Things like playing sport, playing a musical instrument, socializing with friends. So you would say, how long did you play sport when you're in your youth, in midlife, and in late life, for example? So the same questions repeated across these decades. This is the same thing again, just reinforce the point. There are 6 variables here, 3 time periods, specific and non-specific questions, and you just sum the total score across these questions. So the first thing we asked is, do these lifestyle measurements make unique contributions to late life cognition? If you correlate at least these 6 lifetime or lifestyle variables in separate regressions, they all correlate positively, which is not particularly surprising. But with multiple regression, including all 6 of them at the same time, you can ask which of them make a unique contribution. And here are the results. So if we start at the bottom, we found that age had a negative effect on cognitive ability. And this is all people above 65. So this is age from 65 to 88, had a negative effect on cognitive ability. That's not surprising. We know as you get older, your cognitive abilities generally deteriorate. We also found a unique positive contribution of the LEQ's youth-specific questions, which is largely education. So that replicates previous studies that have shown that education seems to be important for late life cognition. But the interesting finding, which replicated the Gao study on independent people, is that midlife non-specific activities, that's outside occupation, made a unique contribution, a positive contribution to late life cognition. So that's over and above education, age, in fact, over and above all these other variables. There's a unique contribution of what you do outside your job in midlife on cognition 20 or 30 years later, using this retrospective questionnaire. So we replicated Gao et al., but we wanted to go further and say, do midlife activities moderate the relationship between cognition and brain? So on 195 of these, we could estimate total grey matter adjusted for head size from T1, T2 weighted MRIs. And what we found was significant interaction between midlife activities and grey matter in predicting cognition. And that's a bit hard to digest, so I'll show you a graph in a moment where I split the lifestyle into those with above median or high levels of midlife activities outside the workplace and those with lower than median activities, just to visualize this interaction. And here it is. So on the y-axis, we've got cognitive ability, Z-scored Cattell, and on the x-axis, we have brain structural health, so total grey matter adjusted for head size, again, Z-scored. You'll see there's 2 colors here. The red are people with lower than average or lower than median midlife activities, and the blue are those with higher than median midlife activities. What you can see in the red group is that the greater your grey matter, the greater your cognitive ability. So a positive correlation in the low midlife activity, which was removed in those with high levels of midlife activities. So in other words, those with high levels of midlife activities, their cognitive ability was independent, or at least less dependent on their brain structural health. So some other factor, possibly functional connectivity, that's what we need to find out, was protecting the group with high levels of midlife activity from their cognition being dependent on their brain structural health. Hopefully that's clear. I think Michael said questions at the end, but if anything's unclear, please do interrupt and I can try and explain better. But that was the evidence for cognitive reserve. So to summarize, we identified a type of cognitive reserve, midlife non-specific activity beyond occupation, which predicted cognition years later over and above education and other current activities in old age. And it reduced the dependency of cognition on brain structure. And it's potentially modifiable by simple interventions, nudging people to be physically healthy, for example, take the stairs rather than the elevator, or to engage in more other activities in midlife. Might be easier to intervene. Now, of course, there are caveats to this study. The foremost of these is that we know that these lifestyle activities are beyond occupation, but with the LEQ, it's difficult to distinguish whether these activities are physical, or the key activities are physical, intellectual, or social in nature. There's simply too few items or too few questions on this questionnaire to try and disentangle those contributions. Oh, sorry, wrong way. The other caveat is, of course, reverse correlation is still possible here. So higher levels of cognitive ability your whole life may have caused healthier lifestyles. So unlike Gao et al., we don't have childhood cognitive ability measured although I suspect that correlates highly with education, and we are adjusting for education here. In any case, we can't adjust for that. But given the Gautel study, it's probably unlikely that this is cognition affecting or lifelong cognition affecting lifestyle choice. Another possibility is that these retrospective questionnaires, your current cognitive ability in old age may affect how well you remember or report what you did early in your life. That's possible, although my pet interest is memory, and while it's true that as you get older, your ability to encode new memories gets worse, so that's why I, for example, forget where I put my keys and where things are, because I'm less able to encode new events compared to when I was younger. But aging doesn't really affect autobiographical memory, that is memory for stuff you encoded when you were younger. That is affected later in neurodegeneration, dementia, but it's not really affected that much in healthy aging. So that's why I think these lifestyle reporting, what you did when you were healthily remembering things early in life, is less likely to be affected by current cognitive ability, though it's still possible. Of course, the gold standard would be to do these prospective studies where you objectively measure lifestyle in middle age and follow the people into old age 20 or 30 years later. And that's, of course, the plan. And that's why it's vitally important to fund longitudinal studies of these aging population. And that's what we're trying to do in CamCAN, but it's very hard to get funding for these long-term studies of 20 or 30 years. Anyway, so cognitive reserve, I think must correlate with some property of the brain if it's not brain structure. If it's not grey matter, what is these lifestyle activities, what are they doing to the brain? So brain function seems the obvious choice, and this brings me to this last study in this first half of the talk. We hypothesized that it might be due to something called functional segregation. So a different Chan published a study a while ago showing that, well, firstly, you can measure large-scale functional brain networks using fMRI during rest, for example. And what you find is that you get networks of regions that are highly correlated within the network, but less correlated across networks. So functional segregation is a summary measure of how separated these networks are. So it's simply the difference between within network connectivity minus between network connectivity. So if you have a cognitive matrix with all regions of the brain, connected to all regions of the brain, and you order that by network, the segregation is basically how well they're connected within a network minus between network connectivity. So that's what segregation is. And what Chan et al. Found was that segregation declines with age, and it relates to cognitive ability, even adjusting for age. So we hypothesized that maybe midlife activities affect functional segregation in old age. 20, 30 years later, your brain just keeps its organization better. So with Peter Rakoff, we looked at that quite recently. And so we sort of pre-registered some analysis, and we're using Chan's system segregation measure that I described just now. We replicated the effect of age on system segregation. So this is the y-axis is a measure of brain segregation, functional brain segregation, and the x-axis is age, and there's a decline. And we found for fluid intelligence, for example, there was a positive relation between functional segregation and fluid intelligence, even after adjusting for linear and quadratic effects of age. So it appears important for cognition, and it gets worse as you get older. But what about midlife activities? Unfortunately, we did not find that system segregation mediated the effects of midlife activities on cognition. In fact, there weren't strong correlations between system segregation and midlife activities in the first place. So not much to mediate. So here's the mediation analysis. There are effects of age and midlife activities and sex, in fact, on fluid intelligence, but there's no mediation by resting state functional segregation, contrary to our hypothesis. So functional segregation does not seem to be a neurocorrelate of the cognitive reserve, at least that part of cognitive reserve related to midlife activities. And we also looked at education. Education didn't affect this either. But of course, there are other possible brain correlates. Maybe it's more specific functional connections. Maybe this summary measure is too abstract. Maybe it's particular connections in the brain that are vital. Or maybe it's structural connectivity or white matter. And I'll come back to that in the second part of this talk, the importance of white matter connectivity. But that's work to be decided, so watch this space for more research in the hunt for the brain correlates of these midlife activities. So let me turn to the last half of this talk, which is concentrating on cardiovascular health. So this is current cardiovascular health, not past cardiovascular activities. And I'll describe 3 studies. The first, highlighting the importance of pulse pressure, the difference between systolic and diastolic blood pressure for the white matter integrity of your brain. Then pulse pressure relating to cognition, and finally bringing all 3 together, so showing that white matter microstructure mediates the effect of pulse pressure on cognition. So the first of these studies was led by Delia Furman, and this looked at white matter hyperintensities in the CamCAN dataset. So you can measure these from flash sequences, for example. So basically, these white areas here are thought to reflect damage to the white matter, and there are automated methods that count the number of these white matter hyperintensities, because they can occur anywhere in the white matter, and the volume of these white matter hyperintensities as well. So for each participant, we have an estimate of the number and volume of these white matter hyperintensities. And with structural equation modeling, we related these 2 variables, lesion volume, lesion number, to measures of blood pressure, diastolic and systolic, and heart rate. And this is adjusted for age. And what we found is that you can see that systolic blood pressure is positively related to lesion volume, but diastolic is actually negatively related to lesion volume. In this context of this full SEM, it's positively related again on its own, but when you take account the other variables, it's negatively related. So this, by the way, this is also adjusted for medication, cholesterol, diabetes, socioeconomic status, etc., and other things not shown in this model. But importantly, the main point is that blood pressure affects your white matter hyperintensities. And more importantly, it appears that what might be key is the difference between systolic and diastolic. So there's a positive loading on systolic, negative loading on diastolic, and this is called pulse pressure, the difference between systolic and diastolic. And that seems to be particularly important for this white matter lesion volume, at least. But does that relate to cognition? And this is work done by Deborah King. And so she took a few more variables. So we have BP here refers to the total or average blood pressure, diastolic plus systolic, but also pulse pressure, which is the difference between systolic and diastolic. She also took BMI, heart rate, and heart rate variability, which is another variable known to be important for cardiovascular health. This is taken from ECG after either high pass or low pass filtering, which is thought to reflect different aspects of heart rate variability. So basically we had 6 measures, and using exploratory factor analysis, we found 3 latent factors. So these are latent factors 1, 2, and 3, but at least one of them seems to relate strongly, 0.9 on pulse pressure. She also took 2 latent factors from some of the CamCAN cognitive measures, which very loosely mapped onto what we call fluid intelligence and crystallized intelligence. So fluid intelligence, like the Cattell test, and crystallized intelligence, which remains more stable with age. And with these 2 measures, you can actually calculate something called the ability discrepancy. This is crystallized minus fluid. And the idea here is that although these are cross-sectional data, you can get a surrogate or a proxy for your longitudinal aging of cognition by adjusting your fluid intelligence by your crystallized intelligence. So the argument here is your crystallized intelligence, which is stable over age, which is positively related to your fluid intelligence. Because it's stable over age, adjusting for that, you kind of adjust for your baseline abilities, what you think your fluid intelligence would be when you're younger. So it's a poor approximation, but it has been argued in the literature as being one way to look at your individual change in fluid intelligence, given what you'd expect from your crystallized intelligence. So your change in old age, I should say, given what you expect from crystallized intelligence, and therefore in young age. So with this discrepancy score now plotted on the y-axis, Deborah found that there's a relationship between this latent vascular factor, which loaded highly on pulse pressure, and your ability discrepancy, but only in people in the old age group. So this discrepancy score increases with age because you're crystallized minus fluid, your fluid goes down. So this becomes positive as you get older. So that's the main effect. But that slope was more positive in the older people. There's significant moderation, even done in a continuous fashion with age. So the bottom line is that pulse pressure seems particularly important for your cognitive ability in old age. And this led to the final study. What is it about the brain that pulse pressure is doing that might mediate this effect on cognition? And this is unpublished work, but what I showed you in the Furman paper was white matter hyperintensive, and these are sort of these are called macrostructural changes. They're very obvious, large changes in the brain from a T1 or T2 weighted image. But we can also use diffusion weighted imaging to look at what are called microstructural changes, so changes in the diffusion of water molecules through white matter tracks. In particular, one summary measure is the peak width of skeletonized mean diffusivity, or PSMD. It's a bit of a mouthful, but basically what you do is you apply a tensor model to diffusion-weighted data. So you estimate fractional anisotropy, so a measure of how isotropic movement of water is through white matter, which will be anisotropic when it's in white matter tracks, and mean diffusivity, which is the total amount of diffusion. You can use the FA to extract a skeleton, what's called a sort of the major white matter bundles in the brain. And then you can just basically look at the distribution of mean diffusivity across all voxels in that white matter skeleton. So you then get a probability or distribution, a density function of the MD across the brain. And this measure, PSMD, is basically the width between the 5th and 95th percentile of those diffusion values. And it's taught to capture the fact that pulse pressure or any insult will potentially have effects at different parts of the brain. You don't know where it's going to affect the white matter, but the more variable that diffusivity is across the skeleton, the more likely it is to be damaged somewhere. And it's been shown by previous groups to be a good measure of a sort of white matter microstructural damage. So we can estimate this a single number per participant. And to start with, Deborah showed that the pulse pressure factor, the latent variable affecting pulse pressure, had a similar profile to cognition. So if you look at this white matter diffusion measure of white matter health, you can see, again, there's an interaction whereby only in old age is there significant dependence of your white matter on pulse pressure. So again, something's happening in old age, which is causing pulse pressure to be important for your white matter health. Like we found cognition. And the last part was to put this together in a mediation model. This is a bit more complicated than I've explained so far, so just forget about speed here. We're basically looking at whether the effect of pulse pressure on fluid intelligence is mediated by this white matter, this PSMD measure, adjusting for age, head motion, etc. And this is actually a moderated mediation because we know age moderates this, but the bottom line is, if you look at the mediation effect on the path between pulse pressure and fluid intelligence, this is the dependency is actually abolished, not significant anymore, when we take into account PSMD. So this is what you call a total mediation of pulse pressure on fluid intelligence by white matter diffusion health. Oh, sorry, diffusion metric PSMD. So again, this measure of white matter health seems to mediate the effect of pulse pressure on cognitive ability. So to conclude, I've shown you two things today. I've argued from retrospective questionnaires that midlife activities are important to maintain your cognition despite the grey matter or structural brain changes that occur in all of us as we get older, and so may be important for cognitive reserve, although we're yet to find the precise brain correlate to this in terms of functional activity, for example. And I've also shown you some data that suggests that current cardiovascular health, particularly in old age, and particularly pulse pressure, may be important to maintain white matter health and thereby cognitive health. So those are two take-home messages. Of course, one missing link here between these two is to ask how lifestyles in the first part affect white matter health in the second part. And that's work we're going to do in future. So that's all I wanted to say. Thanks for your time and attention. I'm happy to take any questions, and this is a summary if you want to ask particular questions. So thank you.
Great, thank you very much, Rik. That's brilliant. Maybe if you go out of presentation mode so we can see if people come on, that would be great. So yeah, if anyone has any questions, so thank you very much. First of all, it was, I think, really great summary and really great pitch for this group. So if you want to, you can ask questions via the chat or switch on your video and your microphone and ask the question.